All right, this is the second mini lesson in protein structures and analysis. So, protein structures is what we're going to be talking about today. And proteins are 3D biological molecules that do the work and form the structures of cells and organisms. Proteins functionally depend on the molecule's size, shape, and chemical properties. So, amino acids are the building blocks of proteins. The combinations of amino acids that make up a protein give the protein a distinctive set of chemical properties. The protein's biological activity results from the chemical interactions it and other molecules make in the cell of an organism. Um, so, if we remember, the structure of the protein will dictate the function, and the amino acids join together to make the structure of the protein, and then the function comes from those interactions. So, the basic structure of an amino acid, it has a central carbon bound to an amino group, which is NH2, a carboxyl group, which is COOH, and then a hydrogen. There is also a side chain or an R group, which varies from amino acid to amino acid. So these R groups are what make each amino acid different. There are 20 standard amino acids, and each of these have the unique R group. And this R group determines size, shape, properties, and function, right? Structure dictates function. Example, glycine is the smallest, where the R group is just a hydrogen, and tryptophan is the largest with an aromatic ring on the R group. So they are classified by that R group. We can see what an amino acid looks like, right? We see the central carbon, the hydrogen, the R group, the carboxyl group, and the amino group. They can be either positively charged and have a basic pH, or negatively charged and have an acidic pH. We can also see polar and nonpolar R groups as well. The chemical interactions of the R groups with each other and the environment will affect the 3D shape of the protein, right? And we all know structure dictates function. So, once we have our chain of amino acids, we have 4 different levels of structure of those proteins. We have primary, secondary, tertiary, and quaternary. So, the primary structure of amino acid sequences in the polypeptide chain is literally just the chain of polypeptides. The peptide bonds form between the carboxyl group of one amino acid and another amino acid, releasing one molecule of water in a dehydration synthesis reaction. The peptide bonds form the backbone of a protein, and the R groups hang from the backbone. So we can see here, right, the polypeptides join together and the backbone hangs off. The secondary structure is generated when hydrogen bonds form between amino acids that are in close proximity of one another. So there are 2 regular structures that these interactions will form. We have alpha helixes, and these are tight coils. They almost look like a spring. And then we have beta pleats, which look like if you fold a piece of paper back and forth a bunch of times. Beta pleated sheets are strands of 3 to 10 amino acids lined up side by side. Tertiary structure is the 3D shape of the polypeptide chain when it's folded. So tertiary structure is determined by interactions of amino acid side chains. For example, hydrophobic side chains may interact in the interior of globular proteins. So if it's hydrophobic, it's going to want to stay away from water, so it's going to go towards the center of the protein, while hydrophilic is going to go to the outside. So these are the larger interactions. At the tertiary stage, proteins become functional And some proteins can just be at the tertiary stage, while others move on to the quaternary folding structure. So, quaternary structure reflects the interactions among multiple polypeptide chains. Not all proteins have quaternary structures. And an example of one that does is going to be hemoglobin. The protein that carries oxygen in our blood has 4 subunits that interact together to form up the blood molecule. That's where we're going to end this mini lesson.